All right, everybody, we're heading into the last set of notes for our molecular genetics unit. Um, looking at the control of prokaryotic genes. So, yesterday we looked at eukaryotic, now we're going to look at prokaryotic because the systems are a bit different. So, as I mentioned yesterday, bacteria have to respond super quickly because their environments are constantly changing all the time. Um, they might have enough of a product, and so they don't want to produce it anymore, or they might need it very quickly. Um, why would they do this? Because they don't want to waste energy, right? They don't want to waste energy producing more of something they already have. So, how do they do this? Well, you have to stop production of the enzymes for synthesis, right? So, if they find a new food, if they find a new energy source, and they need to utilize it quickly, then they better turn things on, right? They better make things go, and they're going to have to interact with that food or that energy source very quickly, use their metabolism. They use it for growth and reproduction, and they will start production of enzymes for digestion of that food source. Okay, so this is an old slide. So feedback in inhibition. So remember how, grab my pen, remember how when we make certain things back in our metabolism unit, right? You make something down here. Here's our product. And the more you make, sometimes it comes up here and it will shut down the enzyme, right? It will shut down that enzyme from working and it will stop the pathway, right? Allosteric inhibitor. Remember, allosteric means a site other than the active site. Um, so it acts as an allosteric inhibitor of the first enzyme in the pathway. But this is a useful production. Uh, uh, this is a wasteful production of enzymes because by the time you do this, you keep making all of these enzymes along the way, and it takes a while to shut down the process. It's still good, but it's not the most efficient. Bacteria or prokaryotes are actually a little bit more efficient. They will just go ahead and the tryptophan will um, the tryptophan will block the transcription of all of these genes. So then they don't even make the enzymes, right? They don't make any of these enzymes where that's even more efficient than just blocking the first enzyme in the path because you still had to make all of those enzymes 1 through 5. So in this case, it saves energy by not wasting it on unnecessary protein synthesis. Okay, so bacteria are actually pretty smart about that. So um, cells vary the amount of their specific enzymes by regulating transcription. We talked about that yesterday with the eukaryotes. So genes get turned on and off. So for an example, if a gene gets turned off, bacterium might have enough tryptophan, which is an amino acid, might have enough of that, so it doesn't need the enzymes built to build more tryptophan. So it would stop production of tryptophan if it has enough. If it encounters a new sugar or an energy source like lactose, then it needs to start making the enzyme that's going to digest that lactose. They can use that energy source, right? They have to start making lactase in order to digest the lactose. So they're turning things on and turning things off constantly. So the way they do this is they use operons. Okay, operons are genes grouped together that have related functions. So basically, if you have a series of genes in this metabolic pathway, they're kind of all governed by the same uh, uh, structures or same features, like the promoters and the operators. So let me grab a pen here. So um, for the promoter, remember the promoter, like in a eukaryote, was the one with the Tata box, right? That transcription factors come and join onto. A promoter for bacteria, in this case, in an operon, it's going to be a single promoter that controls the transcription of all the genes within that operon. So if I go down here to my picture, here's the genes of the operon right here, right? So the whole trip operon is this thing, right? This is the whole trip operon. And this is a gene, 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 and this is a gene. So there's all these groups of genes, but they don't all have their own promoter. They all have this one promoter that sits here in front of those genes. And so that one promoter is there to then control transcription of all of those genes all at once. And that's what the operon is a group of genes that are going to be there excuse me, to make one product where when you have an operon, you're only making a single mRNA. Even though these are different genes, there's only going to be one mRNA made. Okay, so they're basically working as a unit. Okay, so and then you have your operator, which is this little yellow kind of weird section right here. And that's the binding site of what we call a repressor protein. That's where a repressor is going to come up in here, sorry, into here, and it's going to block this at some point. So we'll get to that in a second. Okay, so how can these be turned off, right? So here is the info on the repressor protein. So a repressor, and if you look at the diagram down here, I'm going to grab a different color pen. This is your repressor, right? The red thing right here. And so it is used to bind to the DNA at the operator site, which is going to block the polymerase, and it's going to then block transcription. So look at what happens. Here's tryptophan, right? Tryptophan is this peach-colored molecule down here, and it will fit into here, right? And this is the allosteric site, right? It's not the active site. This is actually the active site right here, okay? So it allosterically binds right here. So now we have the tryptophan, and notice the shape change, right? It was like this, and now it's very vertical. And so the shape changed with the repressor once the tryptophan binds to it, and now this will fit perfectly up into that operator site, that yellow part. And this basically, I mean, look at this big red complex right here, this big red repressor protein. It's just a physical roadblock. It's not going to let anything go by, right? So the polymerase cannot get onto that, and now you're not going to have any transcription. Okay, so it's going to shut down transcription by putting a repressor protein there. So the Operon model here. So again, how they control things. So the polymerase would be going along, reading all these genes, right? And so you would get this one mRNA, and we would make all these enzymes basically off the one mRNA. Okay. So, but if the repressor binds here, if you saw the red circle, the repressor go on there, I'll back this up a little bit. When the repressor goes on there, now the RNA polymerase is not going to be able to get on there. It takes off, and you're going to block transcription, and all of this is going to go away, right? You won't be able to make any more. Okay, so just to finish up down here, the repressor protein turns off gene by blocking the RNA polymerase site, right? That's what the repressor is there to do. So here again is the repressible operon tryptophan pathway, right? So in this case, it's making a lot of tryptophan. So you can see all of the tryptophan being made down here at the end of the mRNA. So this is a pathway where when excess tryptophan is present, it binds to the TRP repressor protein and triggers the repressor to bind to the DNA. So what that means, if I try to break that down for you, is you're making a ton of tryptophan now, right? All these tryptophans are being made. The polymerase is going by, the mRNA is being made, we're making lots of tryptophan. But when we make a whole lot of tryptophan, if we make too much, now the tryptophan is going to start running into the repressor a whole lot more because there's more of it. And so now that tryptophan is going to bind to the repressor, and that is going to cause, I got to get rid of my pen, that is going to cause, now we have the tryptophan activated repressor, and it goes and sits on there, and the polymerase will not be able to get on there, and we will be done making tryptophan for a while. Okay, and so you don't want to just keep making tryptophan. That would be a waste, right? It only wants to make what it needs, and then it wants to shut down that pathway. So that is what we call the repressible operon, right? So so what happens when tryptophan is present? If we have a lot of tryptophan, we don't need to make tryptophan building enzymes anymore. So then we shut down that pathway. Okay, so once we have enough, then the pathway gets shut down. Okay, and that's why we say that tryptophan is an allosteric regulator of the repressor protein. Remember, the allosteric site is not the site that's going to bind to the DNA, it's the other site that's going to cause the conformational change of the repressor so that it can bind to that DNA. Okay, moving on to the next operon, and we're only going to talk about two today. Okay, we just talked about the trip operon or the repressible operon. Now we're going to talk about the lac operon or the inducible operon, which is going to deal with lactose. Okay, so in this case, this is a digestive pathway. So when lactose is present, it's going to bind to the lac repressor and trigger the repressor to release. So in this case, it's a little different scenario. In the lac operon, the repressor is already there. The repressor stays there unless something comes to bind with it, which is going to be lactose. So if lactose binds with this, it will remove the repressor, and then it will start making enzymes to digest that lactose, right? Because you don't want to keep making lactase enzyme to digest lactose if there's no lactose present. You only want it to be generated when the lactose is there. So the repressor is on there all the time unless it runs into lactose. So here, polymerase cannot get on. Most of the time, it will not be able to work. And then when you run into a bunch of lactose, when you run into a big food source, right, sugars, okay, now the lac is going to bind with this. It's going to bind with the repressor, and then conformational change in the protein, and off goes the repressor. Here comes the polymerase. Here comes the enzymes. And now we are going to be making lactase enzyme to digest the lactose. Okay, so the lactose gets made. Okay, so what happens when lactose is present? We need to make lactose digesting enzymes, which is lactase, right? And so, and there's also, looks like there's these other ones down here as well. So we're making, here's the mRNA. We're going to be making the enzymes that are going to help with this. And I don't need you to know those enzyme names. I'm cool with lactase. And then what happens when all the lactase gets digested? Okay, let me see. Nope, I need to go back one. What happens if I go through this? What happens when all of this now, when we remove the repressor and we have the enzyme to now digest all of this, what happens? All the lactose is gone, right? So what's going to happen with the repressor? There won't be any lactose, and now the repressor will go back on, right? So it will come back in here and it will sit there and we're done. Lactose isn't needed anymore once it's digested. We don't need the enzymes anymore, and the repressor will go back on and it will sit there again until it runs into lactose, right? So once if we run into lactose, we need those lactose digesting enzymes, and so the molecule here is going to cause the conformational change, right? This is going to be the lactose inducer, right? And so it is going to cause the conformational change. This is what causes it to release and come off the DNA, and then it can start making the enzymes that are necessary, okay? So again, lactose is an allosteric regulator of the repressor protein, okay? These are the guys that get credit for figuring out how operons work, especially the lac operon. First to describe the operon system, coined the phrase operon. So Jacob and Manad are the guys that did the research on all of this. And for our summary here, the repressible operon is usually functioning in the anabolic pathway, right? Synthesizing end products. And so that's making the tryptophan. And when the end product is present in excess, then the cell is going to allocate resources to other uses, right? And it's going to shut down that pathway. So we make a lot of tryptophan, right? Because that's our repressible one. Once we have too much tryptophan, then the repressor is going to be added on and we're going to shut it down. In the inducible operon, it usually functions in catabolic pathways where things need to get broken down or digested. And so when you run into things, then the repressor gets pulled off. Enzymes are made to do the digesting, right? You produce the enzymes only when the nutrient is available. So only when the lactose is there do we make the enzymes to break them down. And this is just way more efficient, right? So it avoids making proteins that have nothing to do, right? You don't want to make proteins if there's nothing for them to act on. So it becomes way more efficient use of their ATP, use of their energy, and so the Operon system is quite awesome for the prokaryotes, and in some ways is even a little bit more advanced than some things we do. So hopefully that helps you out. That was the last set of notes for this unit. For those of you that like to know what's coming, I can tell you, here's my calendar down here. We're going to be doing a lab on Thursday, Friday. It will run into the first several days of next week. We will review Thursday and Friday, the 18th and 19th, and your next big exam is on the 22nd and 23rd. That'll be when the molecular genetics big exam happens, is on Monday and Tuesday, the 22nd and 23rd. And then we will start getting into this 8-day lab, which is going to be crazy. So that's what we got coming up before spring break here. So not too much longer before we get that. So hope you're doing well. Let me know if you have any problems, okay? See ya.